So for prokaryotes, remember this is a cell type. These cells lack what? They lack a nucleus. There's two domains, though, that contain prokaryotes. What are the two domains?
Archaea and bacteria.
Archaea and bacteria. And most of the time, of course, bacteria is what we've studied the most. It's pathogenic for humans and beneficial. But because of the potential to make us sick, we've studied it the most, right? And remember, do you guys remember when this whole 3-domain system was proposed? It's like within our lifetime. I was like 4 years old. Maybe some of you guys weren't born yet. It was in the 70s, right? In the 70s. So, we're still learning a lot about archaea, right? This new domain, I mean, of course, it's probably been here since way before us, right? But studying it and knowing about it is relatively new. So at one time, prokaryotes were, in our textbook, lumped all in one chapter. But you'll notice that they actually divide it into 2 chapters in your book, right? So you kind of have to go through both of these chapters. And one of the big things we do in science is comparing and contrast, looking at similarities and differences. So I'm going to go through prokaryotes, and so remember I'm talking about bacteria and archaea, but I'm going to focus a lot on bacteria, right? Because that's where our focus has been for a long time. And then the next lecture you'll notice it's titled Prokaryotes and Eukaryotes, because we're going to do a comparison. As we go through the rest of the cellular traits, we're going to compare them prokaryotic versus eukaryotic. Does that make sense to you guys? Okay. So having said that, I kind of moved I do have a separate Connect assignment for prokaryotes and eukaryotes, but when I finish this lecture on prokaryotes, this is only the first half of prokaryotes, right? When we cover eukaryotes, we're going to finish up prokaryotes as well. So I usually suggest that you guys wait until we finish up prokaryotes and eukaryotes to do both the prokaryotic and eukaryotic assignments in Connect. But right now, there's only 2 assignments you've had to do, and hopefully you're on it or getting on it, right? You should have done the chapter 1, right, and the gram stain at this point. If you haven't done the gram stain, get on it, right? And then you got a little bit of a reprieve, and we even get a little bit of break with Mardi Gras, right? Don't let your brains completely turn to mush. So, prokaryotes. They have lots of different sizes, and so we have some terminology that discusses those different shapes. Basic shapes that you'll see of prokaryotes are coccus and bacillus, and those of you guys in lab, yep, that's about as exciting it gets, right? We're going to look at spheres and rods. The rods will be different length and different thickness, and the cocci will be spherical and have different arrangements, but yeah, that's about as exciting as it gets. We don't work with any spirillum. They're too hard to grow and keep happy. Rectangular and square shapes tend to be more of the archaea, not really bacteria. And then as the cells divide, they tend to stay together sometimes and form these characteristic arrangements, like terms that you've probably heard before, like staphylococci and streptococci and diplococci. So we're going to go through each one of these. So for coccus, which would be, remember these are 3-dimensional organisms, this would be a perfect sphere. Beautiful little balls. Coccus is the term that we use for a single organism, a single sphere. Cocci is for multiple spheres. That's the plural term, which is Latin, which is opposite than what most of us are used to, right? When we say I, we mean us, singular, right? Single person, myself. When we say us, we usually mean a group, plural. So basically, if you just remember they're opposite of what we're used to, you'll be A-okay, right? So I is plural, ending in us is singular. So for rods, commonly referred to as bacillus, If you're talking about one, if you're talking about multiple, bacilli, right? That's plural. This should say genus. It could say species, but I left off the species name. So one or the other, I got to fix this slide. Bacillus anthracis, for instance, right? That organism causes anthrax. It's part of its name, its genus, relates to the fact that it is a rod-shaped bacteria under the microscope, right? So imagine a hot dog or a sausage or Mike and Ike's or my favorite, hot tamales, right? Can you imagine that shape? So because it is also a genus name, a lot of scientists like myself just say rod-shaped. We actually don't use the proper term. We don't usually say bacillus. Because sometimes it can be confusing to people. Are you talking about the genus, that group of bacteria? Or are you talking just about shape? But there's another way you can determine if I was writing it, as you see here, what's different about that name as opposed to how I wrote the shapes. It's italicized. Proper names, right, of organisms are italicized. The genus is also always capitalized, but so is any letter at the beginning of a sentence, right? So the context would clue you in as to whether we were talking about a shape or a genus of the sentence. But just eyeballing it, if it's italicized, you know they're talking about a proper name, about a group of organisms. Make sense? So a lot of these terms that we use are proper names for organisms. And again, probably came from the fact that they looked under the microscope, right, and they had this characteristic shape and arrangement. So for Coxobacillus, and I have to say I've never really seen one, these are really small rods, so small that sometimes they even look oval like you see here. And depending on what orientation you're looking at them at, they almost could look spherical. So imagine, right, that hot dog. If you look at it lengthwise, horizontal, clearly looks like a rod, right? But you could flip it and look at it from on top, and it would look circular, would it not? So the same thing with these guys, because they're so short, sometimes they almost look spherical. So, of course, they just smush the two names together, right? They call it Coxobacillus. In lab, the only choices you have, as I've said, is cocci and bacillus. So you're looking, is it a perfect circle? If it's a perfect circle, you're sure about it, it's a coccus. If it's not, it's probably a really small rod. Right? And we've got some really small rods. E. coli, for one. Then we get some really big ones, right? Bacillus megaterium. The name says it all, right? Mega. Right? He's big. Make sense? How would you describe this shape right here? Yeah, it's kind of a rod, but it's got a curve in it, right? It's got a bend in it. Okay, this is known as Vibrio, another one that can be confused because, for instance, Vibrio cholera, right? Vibrio is the genus. Cholera, right, cause a really nasty intestinal infection if you ingest it. And then in this picture, notice this little white line. It has a flagella. Right, so these guys are actually motile, too. And again, a lot of the organisms in the Vibrio genus have this characteristic shape. And a lot of them, too, are motile by a single flagella. This guy, notice a much longer rod with much more curvature to it, right? Many curves, not just one. This is known as spirillum, or spirillia, if you're referring to more than one. And then again, notice in this picture too, this guy is motile. He has multiple flagella. There's a whole bunch of them, balled up here at this end and at the other end. Oh, just the flagella are just touching. It's just like your hair being tangled up kind of thing in this picture. The organism is dead, right? We don't take pictures of them when they're still alive. That's kind of hard because they're moving. So speaking of movement, this next one, your so you have your spirilium. These are your spirochetes or spirochete. These guys are helical, right? Like a slinky or a spiral staircase.
Or spring.
Or a spring, right? And this shape that they have in this very specialized movement ability they have, where they can corkscrew, they can spin through their environment, is due to these black lines that you see in this electron micrograph. You see the black lines? Those are axial filaments. So they're similar to flagella, except they run along the outside of the cell within an outer sheath. This actually gives them the characteristic shape and movement that they have. Scientists have engineered ones that don't make axial filaments. They're just this long, floppy rod. They don't form the helical shape, and they can't corkscrew. They can't move. So these filaments, right, are directly linked to their shape and their movement. The causative agent of Lyme disease. You guys remember what causes Lyme disease? Ticks transmit. What's the causative agent? It's a bacteria. Borrelia burgdorferi. Right? It's a bacteria. Guess what it is? It's a spirochete. Yep. Causative agent is syphilis. Spirochete. Hence why, yeah, we don't work with them in lab. And they're really hard to work with. Same thing with the spirilium. And again with Vibrio too, like we don't want to mess with things like cholera. I'll pass. We still work with pretty pathogenic ones like Klebsiella pneumoniae, right? But again, you'd have to lick the petri dish to get pneumonia, and I won't let anyone do that. So what strong cellular structure helps these prokaryotes hold these specific shapes? It's helping them hold this shape. Why is it they can be spherical and rod? The cell wall. Yeah, the cell wall. With the exception, of course, of those darn spirochetes, right? Because it's actually the axial filament that contributes to that helical shape. But even maintaining that rod shape when they don't have the axial filaments is due to their cell wall. So this structure is believed to play a role in the differences between when we see gram-positive and gram-negative. But it also serves a purpose. Other than shape, it's very important for osmotic pressure. These are single-celled organisms. If water comes rushing in, what happens to our cells if we're in a hypotonic solution and water comes rushing in? They explode, right? They explode. If you're a single-celled organism and you explode, you're dead. So exploding kind of a bad thing, right? We're multicellular. We could lose a few cells. It's all good, right? Single-celled organism, that's death. So you've got to be able to protect yourself. So a cell wall is that rigid wall that keeps them from bursting under osmotic pressure. Well, this is all the same organism. What shape are they? Yeah, they're like all different shapes, right? It's all the same organism. What structure do these cells not have? They don't have a cell wall. And they're what we call pleomorphic. Pleo meaning many, morphic refers to shape, right? So they have many different shapes. There is one genus of bacteria that has this characteristic of no cell wall, therefore it's pleomorphic. Anyone know? No one filled this in because I didn't post it ahead of time. No. Mycoplasmas. Myco, remember, refers to what? Fungi-like, right? Myco is fungi. So see how they're kind of stretching out? So scientists in the beginning were like, oh, maybe it's a fungus, right? Maybe it's mold. Come to find out, no, it's a bacteria, and it's lacking a cell wall, which is why it can't hold the shape. So it just has what? In order to be a cell, what you have to have is a boundary, a cell membrane, right? And sometimes it's called the cytoplasmic membrane, plasma, you see? Mycoplasma, so fungi-like plasmas, they only have a cell membrane, no wall. So, how is it they're not dead? How is it not bursting all over the place? Do our cells have a cell wall? No. Can they resist a certain amount of pressure? How do we do it? Selectively permeable membrane. But I got news for you. Water doesn't give a crap about selectively permeable. It goes. It goes. So we have to have some type of rigidity to our membrane, right? For us, that's called cholesterol. Yeah, you make it. And this is why when you eat animals, you also ingest it. Right? And why some people run into problems when their genetics makes too much of it and they eat too much of it. Right? So then you have to eat less of it and you have to take drugs to take care of the genetic factor because we can't fix that.
So why do we say that exercise is helpful to do that?
That's a good question. Because you don't have the tendency to deposit and have excess when you exercise. When you exercise, your arteries are healthier, your heart is healthier, you can push things along better.
So what's the difference between the two different kinds of isn't there like LDL and
Yeah, there's different levels of cholesterol, and I honestly don't have that memorized. But it has to do with, and think about this, like I said, you make cholesterol. You need it. One, for your membranes. Two, a lot of it is converted into your sterol hormones, estrogen, testosterone, right? So you need certain amounts of the different types in order to be healthy, right? Okay, so for these guys, they have a sterol-like component to their membranes that help them, just like it helps us, resist that osmotic pressure. Mycoplasma pneumoniae causes a very weak form of pneumonia referred to as walking pneumonia. Because people are up walking around and they don't realize how sick they are. A friend of mine in college, we were resident assistants, we were doing rounds in the dormitory, decided to race up 4 flights of stairs. She passed out at the top. She had walking pneumonia. She was up walking around, she felt kind of a little bit under the weather, but not so bad. And then, very strenuous activity like running up 4 flights of stairs. You can't breathe and you pass out. So, if you feel under the weather, right, always go get checked out. You don't know. And in that case, interesting. Mycoplasma, no cell wall, right? Penicillin affects cell wall, peptidoglycan specifically. Did they give her penicillin? No way. They had to give her something else that would affect that bacteria. Because no cell wall, no peptidoglycan, penicillin's not going to interfere with that, right? It's not going to affect those cells at all.
So do they have to give her like an antifungal?
No, it's not a fungus. Remember, it is a bacteria. Just kind of looks like a fungus. So a different antibiotic that works against something other than the cell wall. So, and we'll talk about this later, some of the antibiotics that we use target protein synthesis, cell membrane integrity, DNA synthesis. There's different targets for different drugs. Like, for instance, erythromycin targets protein synthesis. And is it's a common one that they give people if they have penicillin allergy. It's a whole nother ball of wax, where your immune system, penicillin comes in, your immune system goes, oh no, that's bad stuff, right? Which it really isn't, but it thinks so. Right? Make sense? All right. So now, as I said, those are the basic shapes. Sometimes when they divide, they stay together in these characteristic groupings we call arrangements. And cocci have the most arrangements because if you think about them, they're a ball. And in three-dimensional space, they have a lot of choices, right? They could stay in planar space right next to each other like you see in this picture. Or they can go next to and above and below each other. Or it can be random. So in this case, diplo means 2. So they're forming pairs, And again, a genus name, although like Bacillus, Streptobacillus, and Streptococcus, strepto means, what do you see here? They kept going, right? They formed these long chains. So if it's a cocci, it's Streptococcus, of course, or Streptobacillus. And notice again, these are genuses. Although Streptobacillus is starting to fade out, right? And again, they were named after the fact that they were rods and they formed these chains. But then, we're learning more about the organisms and they're getting plunked into different classifications. Although Streptococcus has pretty much stayed together. And you probably know of a pathogen that causes an infection of your throat. Strep. Strep is an abbreviation of the bacteria that causes that infection. It's Streptococcus prurigens. And so I actually use this as a way of telling a story and remembering strepto versus another prefix we use, which is staphylo, right? Or staph. Strepto, I always think about a chain and about how sore my throat was when I had that infection and how forget about swallowing a chain. Or the fact that your throat is kind of like a tube, right? Like a chain. So remember, strepto, chain. Think of strep throat. Think about that chain. Think about not wanting to swallow that chain. They can form packets. And again, this term that we typically use for the packets of 8 in this cube form is Sarsinia, which happens to also be a genus of organisms that tend to form this characteristic arrangement. And then when they cluster, so they're random in three-dimensional space, almost like a grape-like cluster, this is referred to as Staphylococcus. And again, when we say someone has a staph infection, what do we mean? They're infected by a Staphylococcus, usually Staphylococcus aureus. There are pathogenic strains of this bacteria. It can cause really serious skin infections. MRSA, MRSA, is actually an acronym for Myctoline-resistant Staphylococcus aureus. Micticillin is a penicillin derivative. These guys have developed a resistance, right, as the name implies, against penicillin-like drugs. So if you have this infection, even though most staphylococci are gram-positive, we can't give you penicillin. We got to give you another drug. Anyone know what the next one up on the list was? Starts with a V. Vancomycin. So guess what we have now? VRSA. Vancomycin-resistant Staphylococcus aureus. Oh, yay. Y'all, are we going to run out of drugs? Yeah. Yeah. The potential is there. Right? Yeah, it's scary. They're constantly changing.
But they changing the chemical makeup, right? And keep changing.
It's more complicated than that. We're going to get to it. Right? The important things is that we responsibly use antibiotics when they're truly needed. Right? We don't overexpose the microorganisms to them and greater the risk of resistance. And this holds true with taking your entire dose of antibiotic when prescribed, right? Because just because you're feeling better doesn't mean all the bacteria are gone. And if they hang around, they're probably going to be the ones that were slightly resistant and then are going to become resistant. So, and we'll get into the genetics and all the interesting parts of that next unit, next module. Okay, so this is a cool slide I stole from the internet, but it's out there. It has all the cocci. So there's one we didn't mention that's on here, arrangement. So Diplo, again, I will post this for you. You don't have to keep taking pictures. Sorry. I was like, what is she doing? Taking pictures. I will post this one. I will post the one I just finished too, as soon as I get back to the office today, under Module 1. Okay. So pairs is Diplo, right? Strepto means, what do we see there? Chains, right? Tetrad, anyone know what tetrad means? 4. So notice it's a square packet of 4. And actually one of the organisms we work in the microbiology lab has this characteristic arrangement. Sarsinia, what do we see here? Yeah, it's a cube of 8. A square cube, a packet of 8. And then the Staphylococcus, remember this is clustering. Irregular clusters. So this is a good summary slide. I like this one. So we've talked about cell walls. And again, we really focused in on peptidoglycan and bacteria. But archaea, which are also prokaryotic, do also have a cell wall. And depending on what type of molecules that cell wall is made out of, they will stain differently just like bacteria. They'll either be gram-positive or gram-negative. So although archaea do not contain peptidoglycan, because remember PTG, abbreviation for peptidoglycan, is unique to bacteria. You guys remember what peptidoglycan is made out of? What type of molecules? It's sugars, right? Alternating sugars linked together by what? So what makes up protein? I heard protein. Amino acids, right? So it's sugar and a little bit of protein, basically. And how it responds is it shrinks up. So there are some archaea, their cell wall, is made out of pseudomurine, so it's similar to murine, or complex polysaccharides. That's what? Sugar. What is peptidoglycan? Mostly sugar. Similarity there, right? So guess what? They respond similarly. It shrinks up. It traps in the crystal violet iodine, and they stay purple, gram-positive. Other archaea, their cell wall is made out of protein or glycoprotein. So glycoprotein means it has a little bit of what? Sugar. But not enough, clearly, right? Doesn't shrink up, doesn't trap in the crystal violet, so they end up being gram-negative. So notice I have here thin peptidoglycan or no peptidoglycan. So mycoplasmas. Cell wall? No. Peptidoglycan? No. What would they stain? Gram-negative. Nothing to trap in the crystal violet iodine. If you can't trap it in, you're going to end up being negative. So our cells, if we were to stain them, they'd be what? Gram-negative. Why? No cell wall. Nothing to trap it in. Doesn't even matter that it's not made out of peptidoglycan. It's not even there. No cell wall. Plants. They have a cell wall. It's cellulose. It's a sugar. I have to say I haven't experimented with this one. Might be my new thing for this summer with the kids. Gram stain some plants. See what happens. I'm gonna, as a scientist, I'm gonna hypothesize, right? Anyone know what cellulose is made out of? I almost said it. Glucose. Glucose. Which is a what? Sugar. Maybe positive. Who knows? To experiment and find out. Of course, why would we not do that on plants? Doesn't really tell us much. We know they have cell walls, right? Yeah. And probably they're all going to come out because they all have the same cell wall composition. They're either going to all be gram-negative or all be gram-positive, right? What good does that do for us? Nothing. Where with bacteria, right, or archaea, there's about a 50-50 chance it'll be one or the other. If you don't know what your organism is that you're dealing with, and we can narrow down the possibilities basically in half, that would be beneficial, right? So the gram stain is very widely used. It's very helpful because it tells us something about the organisms and really helps us narrow down the possibilities basically in half. About 40% of all known bacteria are gram-positive. The other 60% are gram-negative. So Compare the structure of gram-positive and gram-negative bacterial cell walls and explain how these differences between it contribute to their differences in the gram stain. So I think we're pretty good on this one, right? Okay. And we can hypothesize. We just did that, did we not? Right? Given what we know, right, we can kind of guesstimate as to what the result may or may not be based on the properties of a cell. Right? Does it have a cell wall? What is it made out of? Right? How might it respond under these conditions? Good critical thinking skills, right? So does the gram stain have anything to do with charge? When I say gram positive and gram negative, am I talking about charge?
No.
No. I'm talking about a result, right? Did it stay purple or did it not and come out pink? Charges are involved, though, at the molecular level, right? On how it works, right? Gram stain works because we're using positively charged basic dyes, crystal violet and safranin. They're attracted to the negative components inside the cell, right? So they go inside that cell. Make sense? Okay. So charges are involved, but when we say positive and negative as a result, that's what we're talking about, a result. Did it stay purple? Did it not? And then, of course, for bacterial cells, it's that peptidoglycan in the cell wall that's going to be contributing factor. And we've already hypothesized, right? We know that we are gram negative. It would be pink. So this is my pretty picture, right? So remember, for bacteria mainly, right, we're talking about a very thick layer of peptidoglycan if it comes out gram positive. If it's gram negative, it probably has this structure. Notice I said probably because remember, mycoplasmas just have a membrane, right? They don't have this peptidoglycan layer and they don't have the outer membrane. But for the most part, when you say gram-negative bacteria, you've got this cell membrane, a very thin layer of peptidoglycan, and this very prominent outer membrane, which is bad news for us because it's toxic to us. Gram-negative infections are bad news, which is why, again, right, a doctor would want to know, what do you got? Is it gram-positive? Is it gram-negative? How am I going to approach this? Mm-hmm. It's how we make the right choices.
So, say for instance, if they're not sure, you know how sometimes they say, well, let's put them on a cephalosporin. Why do we do that test first instead of something like on chemical?
Sulfur drugs. Sulfur drugs, I think, are classified as broad spectrum, which means they affect both gram-positive and gram-negative. So if they're not sure what it is, we're just going to, atomic bomb them, basically. Which is not good news for our body and our good guys, right? The good guys in our gut get kind of disrupted when we atomic bomb them. It's not good news. So it's better to be specific if you can. But when in doubt, atomic bomb. So what's the composition and function of the prokaryotic plasma membrane and its limited internal membrane structures found in prokaryotes? So we know it as a barrier, right? It's actually what defines the cell as being a cell. In that membrane, as I said, hapnoids, these are your sterile-like molecules that add rigidity. It is a lot of times referred to as your fluid mosaic model. Fluid meaning it's what? It moves, right? So imagine those phospholipids and all those proteins embedded are like icebergs floating through a phospholipid sea. Right? They're constantly moving and changing. It's not stuck. Unlike a mosaic, right? You break up a whole bunch of tiles, you slap them on the wall and make a pretty picture, and that's it. You glue them on. In this case, mosaic, meaning it's made up of many different parts to present the whole membrane. Right? So that it all comes together to form this beautiful structure, this membrane, all the individual parts. But it's not stuck. It's dynamic. It's flexible. It's constantly changing. So for the structure, right, most of us have seen this before, right? This is the common little drawing that we depict for the molecules, but what you have up here is a phosphate, which notice it's negatively charged. It's phosphorus and oxygens. Phosphorus is a little greedy, takes more of the hydrogen atoms. Or actually, no, it's the other way. Electrons. Oxygen is electron greedy. Keeps more electrons, so it becomes negatively charged. The important thing with that, though, is if you have a charged molecule, it's hydrophilic. Hydro means what? Love, water, right? Hydro means water, philic means love. These molecules love water because what's the problem with water too? It's oxygen's greedy too, right? It pulls most of the electrons towards it and leaves the hydrogen mostly what charge? Positive. So it's what we call polar, right? Where you got opposite ends, positive and negative. So water interacts with other charged molecules. When you get down here, do you see any charges on the fatty acids?
No.
They're all about sharing equally down there. Carbon and hydrogen, they're really good at sharing equally. They're not greedy about electrons. So you don't get any of these charges. And because you don't have any of these charges, it and water don't mix, do they? Fatty acids, oil, water, don't mix. This is where we're hydrophobic, fearing water. Basically don't get along with water. So these molecules naturally form these barriers, these membranous barriers. If you just take phospholipids and throw them in some water, they're going to orient themselves so all the phosphate heads are sticking out and all the fat is in the middle, away from the water. Right? They're like, I'm sticking with my buddies. We all agree about sharing. Right? On the outside is all those phosphates that are okay with water. Right? They interact with it well. So when you add oil to make salad dressing to a water-like substance, for instance, vinegar, right, and you shake it up, all you do is create tiny little oil droplets within that vinegar, right? And over time, what happens? They all run back to their buddies, right? They separate out and form those two distinct layers. So why doesn't all salad dressing do that? Nah, they screw with it. They hydrogenate it. They add hydrogen atoms to suspend the oil within that solution. Right? When you make them really tiny, that's called emulsification. You do this in your body all the time. You use salt to do it. You use your bile salts that's produced by your liver and stored in your gallbladder. That's how you break up your fats, make them small so the enzymes can get in there and break up those pieces. Okay? When you buy things that have been hydrogenated, they've added hydrogen to keep it solid, to keep it from separating into those layers. They basically made it what's called a saturated fat. Right? Saturated fats are what at room temperature? They're solid. Right? Because they get all this hydrogen, whether it's naturally or somebody put it there. Unsaturated, on the other hand, are what at room temperature? Liquid. Those are your oils. Does temperature affect?
Yeah.
Oh yeah, because what can you do with a saturated, right? All these hydrogens, really thick molecule, right? Sticking together, staying solid at room temperature. What happens to them when you heat them? They melt. They liquefy. You affect these bonds. Why is it that unsaturated is liquid at room temperature? They have less hydrogen. Notice, doing a little bit of extra sharing here. The carbon atoms are sharing a little bit more, so they don't have room for another hydrogen buddy. And in that, it gives this molecule flexibility. Hence why it can be liquid at room temperature. But of course, temperature affects what happens to them if you were to cool them down. Yeah, they wouldn't flow as well, right? Get kind of thick. So our membranes are made out of this stuff, right? Fatty acids, right? Hydrogen and carbon. It's either saturated or unsaturated. Is that going to be affected by the temperature of the environment the organism lives in? Absolutely. Which is going to dictate what? So, are you saturated because the environment is hot, or were you saturated to begin with and you got to live in the environment because it's hot? Yeah, it works that way. You are what you are, and you stay alive because the environment lets you. What do we call that? Adaptation. More specific. Natural selection. Survival of the fittest. So evolutionary-wise, it kind of makes sense that we see these differences between archaea and bacteria. Because what do we say about the archaea? What type of environments do they like? Extremes, right? So we're talking really hot, really cold, and really salty. Does your membrane have to account for that? Absolutely. Otherwise, you're going to die. So for bacteria, they're going to have bilayers, right? Where there's two distinct layers, right? You see the separation there between... Some archaea are like that too, but they've got these additional branches. You see those little knobs sticking off? For bacteria, it would just be one ziggy line, no branching, right? So some archaea have adapted by having those branched fatty acids. The connection here, too, between the phosphate group and the fatty acids is different. Ester, archaea have ester instead of ether. I can't even keep it straight, right? I don't expect you guys to memorize that. What's the important take home here? It's different. And because it's different, they can survive in different environments, right? They can also form monolayers. So notice here, no separation between the layers. It's all one. Why would that be beneficial? Well, think about it. If you have more than one layer, what could happen? They could come apart. They could slide, right? If you're stuck, right? So women can understand this usually, although I didn't go that route. What do they typically do with the wedding band and the engagement ring? You solder them together because that rubbing over time wears them thin and makes them brittle, right? So you stick them together. So in this case too, right? If you stick them together, they're less likely to come apart or wear apart or shift, right? In those environments. They can even, and I don't know why, this is kind of cool though, they can change the diameter, how thick the membrane is. They can cause the carbon atoms to form circular structures, right? So you see these little houses, right? So it's actually wrapped around and formed a little circle. So that made it shorter in that section. Do you see that? Cyclic, pen referring to 5. There's 5 carbons. That's why it looks like a little house. 5 points of the 5 carbons. So, makes sense to have a membrane, right? To find the inside of the cell and the outside. So that's a very clear functionality, right? In controlling what gets in and what gets out. But look at here, y'all. That's membranes inside. It's not chloroplast. It's not a mitochondria. But it's very similar to these guys in that you've got these stacked membranes inside. So anybody remember from biology class, why do you have those stacked membranes in a chloroplast or a mitochondria? It's the energy, right? They're pumping hydrogen atoms from one side to the other. They're creating a chemical gradient to drive energy. That's what these membranes are doing because, think about it. If you're a prokaryote, if all you have is the cell membrane, that's not enough. Right? You've got to have more membranes to create that gradient to generate energy. So what's embedded in that membranes of the mitochondria and the chloroplast? Proteins. Just like the outside membranes, right? It's those proteins that make up a system we know as the ETC, electron transport chain. Ooh, starting to come back, huh? So what's the function? Energy production, right? To create that barrier for the electron transport chain. So this is it. Wow. Today I actually did this one good. So we're going to pick up on the rest of this. So the next objective, we're going to be looking at different characteristics, comparing and contrasting them. So definitely for homework, see if you can fill in this table. Remember, this is the type of homework I don't collect, right? It is to your benefit to read ahead, prepare ahead of time, bring it with you to class.